Now we'll talk about red cell antibodies, part 1. And I included a picture of Piggy with her little dolphin teeth. So our learning objectives for this talk, we're going to discuss the broad categories of red cell antibodies, and then we'll dig into the ABO system. We'll talk about the genes, antigens, antibodies, and rules of compatibility. We'll discuss briefly the 3 main lab tests that we use for pre-transfusion compatibility testing: the type, the screen, and the cross-match. And we'll also spend a few minutes on type discrepancies. And then finally, we'll discuss the Lewis system, which has some similarities with the ABO system in that they're both carbohydrate blood group antigen systems. So, the way I think about just grouping categories of red cell antibodies is, first of all, we have the ABO category. That's the most clinically significant because an ABO incompatibility in a red cell transfusion can cause an acute hemolytic transfusion reaction. Then I also think about, okay, there are non-ABO antibodies. And there are, broadly speaking, 2 categories of those. One, you have your IgGs, and we usually call those clinically significant. So clinically significant in this context means this antibody can cause a delayed hemolytic transfusion reaction or hemolytic disease of the fetus newborn. And then we have IgM, non-ABO antibodies, and those are generally clinically insignificant. They don't cause delayed hemolytic transfusion reactions or HDN. So one way to think about it is you have alloantibodies and you have autoantibodies. They could be IgG or IgM. Broadly speaking, of course there are exceptions, but broadly speaking, you have your IgMs that are alloantibodies are basically your anti-M, anti-N, and Lewis antibodies that we'll talk about in this talk. And then essentially, when you look at the panel, basically all the others are alloantibodies that are IgG. There are exceptions to that broad statement, but in general, things like your Rh system, your Kells, KID, Duffys, and then S's, those are IgG alloantibodies. Then, of course, you could have autoantibodies, right? You can have an IgG that's a warm autoantibody that's generally clinically significant. And then you can have a cold autoantibody that's IgM that's generally clinically insignificant. So a quick review of ABO. I'll go through this quickly because you probably know it by now. You could have type A, type B, depending on if you have the A antigen or the B antigen, respectively. And you could be type AB if you have both, or codominant. And you could be type O if you have neither A nor B. And of course, you make the anti-A or anti-B against the antigen that you lack. So if you're type A, you naturally make anti-B. If you're type B, you naturally make anti-A. If you're type AB, you don't make either one of these. If you're type O, you make both anti-A and anti-B, along with an IgG that recognizes both A and B. It could recognize either A or B. And of course, genetically, you can be type A if you're homozygous for the A gene, or if you have one A gene and then one O gene. You could be type B if you're homozygous for B, or similarly, B and O. There's only one way to be AB, and that's if you have an A gene and a B gene codominantly expressed. And then there's only one way to be type O, and that's if you have 2 of these O genes. Okay. So the books often show you the entire chemical structure. I frankly don't think that's that high yield. I don't think it's worth knowing the exact chemical structure, so I just put goofy little boxes as a representation of the chemical structure. But what is a little more important is how these antigens are formed, how these structures are formed. So you start out with a type 2 chain, which has a chemical structure that's not worth knowing, that's on the red cell. And then most of all humanity has the H gene. So most everyone has the H gene, and that encodes a fucose, right? That encodes a fucosyl transferase that adds a fucose to the type 2 chain, and then you have the H antigen. Okay, so the H antigen. If you're type O, regarding A and B and H antigens, you stop here. You stop at the H antigen if you have no A gene or no B gene. If you have an A gene, however, that encodes for an enzyme that adds N-acetylgalactosamine. So N-acetylgalactosamine. You add that to the H antigen in order to make the A antigen. If you have the B gene, that encodes an enzyme which adds galactose to the H antigen, and that gives you the B antigen. And so if you're type AB, you do both of these. And again, if you're type O, you stop here at H. So again, you don't have to know these chemical structures, but just to show you what that previous diagram is saving you the stress of looking at, you have a type 2 chain. Most everyone has an H gene, so it adds fucose. Then if you have an A gene, that adds an N-acetylgalactosamine. And then if you have the B gene, that adds a galactose. And how common are these ABO types? O is the most common in the US and in general across the world. Type A is the second most common at about 40%, so type O about 45%, type A about 40%, B 11 to 12%, and then AB about 3 to 4% in the US. And then the three main tests that we do the ABO type, and that's just a description. It answers the question, what is the patient's blood type? Do they have the A or B antigens? Do they have the anti-A or anti-B antibodies? The antibody screen answers the question, does the patient have non-ABO antibodies? Does the patient's plasma have non-ABO antibodies? And we'll get into that in a little more detail. And then the cross-match is specific to a particular unit. So before we take a unit of red cells and issue it, we do a cross-match, and we mix a little bit of red cells from that unit with the plasma from the recipient, and we ask the question, is this unit compatible with that patient? So it's a kind of an in vitro transfusion before we issue the unit. And the whole point of doing ABO type, the primary reason is we want to prevent an acute hemolytic transfusion reaction. And again, acute hemolytic transfusion reactions can be caused by an ABO incompatible red cell transfusion. And a few words on serology, in case you haven't already thought about this, that basically in most serologic testing, we have a known substance, and then we're looking for characterizing an unknown. So, for example, if you mix an antigen, known antigen with patient plasma, we're looking for the cognate antibody. If we have a reagent antibody, then we're looking for the presence of the cognate antigen. And so if the endpoint is reactive, when we mix this known and an unknown, then that means the unknown has the cognate. So again, the type tells us which antigens are on red cells, and so what is the recipe? It's worth remembering the recipe for these lab tests. What is mixed with what, right? If you could remember that, then you could reason through the rest. So in an ABO type, we're mixing anti-A with patient red cells in one tube. In another tube, we're mixing anti-B with patient red cells. And so, of course, we're asking the question, does the patient have the A antigen? Does the patient have the B antigen, respectively? And then we're also doing, in two other tubes, which antibodies are in patient plasma. So we're mixing reagent A red cells, right? Reagent A red cells with patient plasma to look for the presence of anti-A in the patient's plasma. And in another tube, we're mixing reagent B red cells with patient plasma to look for the presence of anti-B in patient plasma. So just to show this in a table and what the results could be, if we have the anti-A reagent and that's negative with patient red cells, and the anti-B reagent that's negative with patient red cells, this is telling us the patient does not have the A antigen and does not have the B antigen. So the going idea is that's probably what? It's probably type O, right? Furthermore, if we do testing with anti-A and patient red cells and that's positive, if we see agglutination, that's a good signal the patient has the A antigen. And if it happens to be negative with anti-B reagent, then we could deduce the patient is type A. It's type A because they have the A antigen and not the B. Conversely, if we test with anti-A and it's negative, they don't have the A antigen. We test with anti-B, it's positive, they do have the B antigen, so the patient is type B. Finally, if it's positive for both, the patient is type AB. So all that is called the forward typing, antigen typing, front typing. Those are all synonyms of using reagent antibody, reagent antisera to look for the presence of the cognate antigen on patient red cells. Then we do the reverse typing or the serum typing, back typing. Those are all synonyms for scrutinizing what is present in the patient's plasma, what's present in the patient plasma. So the reagent, of course, we need a known antigen source. We need known red cells. So we use reagent A1 red cells and reagent B red cells. And so when you have positivity with A red cells, that's telling you the patient has anti-A in their plasma. And if you see positivity when we use B red cells, that's telling you the patient has anti-B in their plasma. So having both anti-A and anti-B, that means the patient is type O. So we do antigen typing and the serum typing in order to make sure that they're consistent. On occasion, they could not agree with each other, and that's a type discrepancy, and we'll talk about that a little later. So that first combo is no A or B antigens, and the patient does have anti-A and anti-B, that's type O. If the patient does not have any anti-A in their plasma, but does have anti-B, then that's consistent with type A. If they have anti-A but not anti-B, that's consistent with type B. And if they have neither anti-A nor anti-B, then that is consistent with type AB. And then for the rules of compatibility, as we discussed in our intro to transfusion medicine and transfusion reactions 1 talk, if you're type O, remember the recipient is making anti-A and anti-B, so they can't see the A or B antigens. Don't infuse them with either the A or B antigens. So type O recipients can only get type O red cells. So if you're a type A recipient, then you make anti-B. So you have to avoid being infused with B antigens. So you cannot get B red cells or AB red cells. You can get your own type, right? Type A, and you can get type O red cells. If you're type B, you make anti-A, so you don't want to receive any A or AB, but you can certainly get your own type B and type O. And finally, if you're an AB recipient, you don't make either anti-A or anti-B, so you could receive any red cells. And of course, a puggle is what combination of breeds, what 2 breeds makes a puggle? It's of course a pug and a beagle, This shows up on the boards every 3 or 4 years. And then a brief word on type discrepancies. We'll mention certain causes of type discrepancies, including A subgroups, acquired B phenotype, and a patient who has undergone a bone marrow transplant is in the transitional period. So A subgroups, so some type A, and especially even more represented in type AB, some A people and some AB people are A subgroup, roughly 20% prevalence. And the most common of these A subgroups is so-called A2. So A2 is the most common of these. And among the A2s, about 1% of A2s make an anti-A1. About 1% of A2 people make anti-A1, and about roughly 25% of people who are A2B make anti-A1. So the most common person that makes an anti-A1 is your A2Bs. But keep in mind that this anti-A1 is different from anti-A, different from the anti-A that you would see in, say, a type B person or a type O person, in that anti-A1 is usually clinically insignificant. There are exceptions, but in general, clinically insignificant. And the reason for that is because this anti-A1 usually only binds at room temperature or cooler temperatures. We could find it in the lab at room temp, but it doesn't bind in the body at 37 degrees. So in general, again, there are exceptions, but in general, it's okay to transfuse A1 containing red cells, assuming your cross-match is compatible at 37 degrees. So if you have an A2 or A2B patient with an anti-A1, then how will that be manifest in the lab? How will you know this? Your ABO typing will show some low-level, unexpected anti-A1 in their plasma, right? So if you see that, and I'll show you an example of that, what you should do next is so-called lectin typing. So you should use this lectin called Dolichos biflorus to see if the patient is indeed A1. If it's positive, if you see agglutination, that means the patient's A1. If it's negative, it just tells you the patient's not A1. It doesn't specifically tell you the patient's A2, but if you were to wager, that would probably be correct, but it's not evidence that it's A2, it's just a negative reaction with Dolichos biflorus just means the patient's not A1. So if positive with Dolichos biflorus, that's telling you that the patient is A1. If negative, then the patient is not A1. So I like dumb memory aids, and one such memory aid is that A1 is the best. You know, like you see A1 dry cleaners or A1 steak sauce. What they're trying to communicate to you is that it's the best, right? So if you change the best to the best, you know, DA best, then the DB I think of as D as in Dolichos, B as in biflorus. So A1 is the best, A1 is DB. So if you see positivity with DB, then that's telling you it's A1. So Dolichos biflorus, DB, positive, tells you it's A1. I told you it was dumb. And then moving on to acquired B phenotype. This happens when essentially there's any sort of interface between E. coli and red cells. And so how does this happen? So you could have underwent colon surgery or injury, you could have colon cancer or bacteremia of any etiology. And so how does this happen exactly? Well, the E. coli contains D-acetylase, right? D-acetylase, which turns N-acetyl, right? N-acetylgalactosamine into galactosamine. So it removes the acetyl group of the galactosamine of the type A antigen, and that is very similar to galactose. Because remember, galactose was the defining sugar of the B antigen. So you can see how if you use an anti-B typing reagent, there can be some positivity with galactosamine. So how you would see this on your typing results is if you have an A patient with strong 4+ A antigen expression, A antigen result, that has a low level unexpected B antigen. So if you have a little bit of B antigen that's unexpected, then this is a hint that the patient has acquired B phenotype. Go look in their history to see if any of these are true. If they have colon cancer, colon surgery, or injury, bacteremia. And the main thing to keep in mind here is the acquired B phenotype is in an A patient, right? So they still make anti-B. So you don't want to transfuse B red cells. And probably the most common cause of a type discrepancy is a patient that has underwent a bone marrow transplant and is in the transitional period. So, of course, if the patient cleanly types, meaning if they type either as their pre-transplant original self or as the post-transplant, everything's been engrafted donor typing, then of course use that type. But what I'm speaking specifically about is the transitional period. If the patient has a type discrepancy between those two, right, between their original patient self and the donor, then use blood products that are compatible with both types. Okay, use blood products that are compatible with both the donor and the recipient. That's consideration number one. And consideration number two is, if possible, save on O red cells and AB plasma and platelets, right? We try to not use O red cells unless we're giving it to a type O patient, or we try to not use AB unless it's an AB patient. And for example, if you have an A recipient and a B donor, of course you have to use type O red cells because there are no other red cells that are compatible with both type A and type B. And you got to use AB plasma because you need to avoid anti-A in the plasma and anti-B in the plasma, so you can only use AB. So that's an example of you're locked in, you can only use O and AB. But there are different circumstances. For example, if you have an A recipient and then an AB donor, then you can use type A red cells, right? You could safely use type A red cells because neither the recipient nor the donor make anti-A. But then we are locked into using AB plasma because, again, we have to avoid anti-A and we have to also avoid anti-B. Another example is if you have an A recipient and then an O donor, we're locked in, we have to use O red cells because no one else will be compatible with the O, but we can use A plasma, right? Because all we have to do is avoid some anti-A. So we could use type A or type AB successfully for that patient in that transitional period. Okay. And then a few words on Bombay. I told you before that most all of humanity has an H gene, and they're able to add that fucose to the type 2 chain to make the H antigen, right? Well, very rarely you could find someone that does not have an H gene, and thus they have no H antigen on the red cells. And if they also lack the secretor gene, then they also don't have any H antigens in their secretions. So this is important because if you're Bombay and you lack the H antigen, you naturally make an anti-H antibody, which is clinically significant. So such patients are only compatible with other Bombays, meaning other patients that are H negative. They would type as type O in the lab because they make anti-A and they make anti-B and they lack the A and B antigens. So the main way this is discovered is you give the patient type O red cells and they have an acute hemolytic transfusion reaction. You do further testing and you find out, look, yikes, they're Bombay, and we need the very rare Bombay red cells for them. So that's Bombay. Para-Bombay just means they have a secretor gene and they have no H gene. So they have the secretor gene but no H gene, so they do have some H in their secretions. This will make more sense when we talk about Lewis and how secretions can play a part in the ABO antigen expression. So they are positive in their secretions. And so in general, they usually have a weak reacting or cold/non-reacting anti-H. So they're not typically in that dire of a circumstance compared to the Bombays who have a clinically significant anti-H. So the para-Bombays can often be successfully transfused with cross-match compatible O red cells. Oh, this is Piggy demanding something. She's trying to charm me into, I don't know, getting her some food, getting her something to play with. So we talked about the type, and now let's move on to the screen. The antibody screen is this recipe. You mix patient plasma, right? Because we're asking what antibodies, it's an antibody screen, right? What antibodies are in patient plasma? So we need some known red cells to mix with the unknown patient plasma. So the known red cells are typically from 2 type O reagent red cells. And we have one of those per tube, and the key here is that we have known extended phenotypes, right? We know if these reagent red cells have, let's say, the D antigen or not, the big E antigen or not, the big K antigen or not. And it's also worth keeping in mind that we're using type O reagent red cells because we want any reactivity to be just due to antigens outside the ABO system. If we were to use some A or B or AB reagent red cells, we would have some reactivity if the patient happened to be making anti-A or anti-B. So we're interested in non-ABO antibodies. Thus, we want O red cells only for the reagent red cells. So you've probably seen these by now. Each row represents one reagent red cell. So this store-bought reagent red cell has the D antigen, has the big C antigen, does not have the big E antigen, does not have the little c antigen, but does have the little e antigen. So wherever there's a plus, that means the antigen is present. If it's 0, it's not. And so the idea is we mix patient plasma with this red cell, and then we can look at negative reactions. So if a reaction is negative, then we can rule out that the patient does not have antibody to the cognate antigen. So for example, if this yields a negative reaction, the patient does not have anti-D, does not have anti-big C, we can't comment on big E because that antigen wasn't there. So we start with the negative reactions, and then we rule out, and then we next look at the positive reactions and then rule in. And we look for a pattern that is explained by either one antibody or several antibodies that yield that reaction pattern. So we mix patient plasma along with reagent red cells, type O reagent red cells with known extended phenotypes, and then we also add AHG, anti-human globulin. And I'll show you a picture of why we do this. We do this in order to bridge the gap, right? We want the endpoint to be visible agglutination. And if we're working with IgGs, or we're trying to detect an IgG, then an IgG by itself onto a cognate antigen, all that's going to yield is a red cell that's covered in IgGs. There's nothing visible that we can see. There's nothing that changes in that sense. We can't see the tiny little antibodies. But if we include an anti-IgG that can recognize any kind of IgG, it just recognizes the FC portion, then that will bridge the gap. That will induce visible agglutination. A positive screen tells you that the patient's plasma contains antibodies to some non-ABO red cell antigens, right? Further testing will determine the specificity, right? The 2-cell screen just tells us positive or negative. If it's positive, we have to do more of those reagent red cells in the panel until we determine the specificity. So the details of that will be covered in a panel party session, and also it's covered by doing the real-life clinical workups of these panels and the antibody reports. Whereas a negative antibody screen means that the patient's plasma does not contain antibodies to non-ABO red cell antigens. And what is the purpose of doing the antibody screen and the antibody panel? Well, first we're going to look at the history in addition to the current screen or the panel, right? And then we're going to use our knowledge of which antibodies are clinically significant. And if the patient has a clinically significant antibody, either in their past history or presently in their current sample, the whole point of this is we want to find some antigen-negative red cells. So I mentioned the history check. Why is that important? In certain cases, the antibody screen is negative because there is no antibody to detect. It's below the limit of detection. So the history is important because you might have an antibody screen that's negative today, but it was positive in the past, and we still need to respect that no matter what the result today. So keep in mind this testing algorithm. We can do an ABO typing as a description of do they have A or B antigens, anti-A or B antibodies. We can do the antibody screen, and then if that's negative, we just worry about the ABO compatibility. If it's positive, we do further testing in the panel, and we find out the specificity. Once we have the specificity, and if it happens to be a clinically significant antibody, then we will so-called respect that antibody by finding antigen-negative red cells. So red cells that lack that cognate antigen, and then we'll do the cross-match. So in the cross-match, what do we mix together? We mix together patient plasma with a small volume of red cells from a specific unit of red cells, right? This is right before we issue that unit. And it's sort of like an in vitro transfusion. We want to make sure if there are any troubles, we discover them in the lab, not in the patient. And if the cross-match is compatible, then we could issue that unit. And you might argue, well, isn't the cross-match redundant? I mean, we already did the ABO typing, we already did the antibody screening. Sure it is, but it's redundant to some degree, but it's also a fail-safe that could catch clerical errors, and it could catch rare antibodies that the patient may have to antigens that are not on the panel. A cross-match can pick up those. And then moving on to Lewis, remember from Revenge of the Nerds, this is Lewis. That's what we're going to be talking about. And of course, this is Gilbert and Lewis. Big picture, you got to remember that Lewis antibodies are clinically insignificant. In general, they're clinically insignificant IgM alloantibodies. Yet they are relevant to PIMP questions in the boards because you could be asked how the antigens are made, and you could be asked factoids about Lewis antigens and antibodies that we see in pregnancy and in neonates. So Lewis antigens are a little bit different from your typical red cell antigen. Lewis antigens are synthesized by intestinal epithelial cells, so they're not intrinsic to the red cell. They're passively adsorbed onto the red cell. And antigens sometimes will drop their expression levels during pregnancy. Sometimes they'll go from slightly positive to actually negative. And if that were to happen, sometimes it will result in a transient antibody. And it's also worth remembering that Lewis antigens are absent on fetal, neonate, and cord red cells. So Lewis antigens are not yet expressed on neonates. And my dumb memory aid is that Lutheran, so Luther and Lewis are lower and lowest. So Luther and Lewis are lower and lowest, meaning Lutheran antigens are very poorly expressed on neonatal and fetal red cells, whereas Lewis antigens are not expressed at all on fetal and neonate red cells. So that's respectively lower and lowest. So how are Lewis antigens made? This is worth remembering because I could ask you, given a patient's genotype, what's the phenotype? Or given the patient's phenotype, what's their Lewis genotype? So if you have a type 1 chain in your secretions, which is very similar to a type 2 chain that's on the red cells, but just a slight bond difference. If you start with that in your secretions and you have the Lewis gene, right? So you have a Lewis gene, then you make the Lewis A antigen, okay? So you add on a fucose to the type 1 chain and you make the Lewis A antigen. Now keep in mind, you have a Lewis gene or no Lewis gene, right? It's different from other antigen systems in that this is not a Lewis A gene. So there's not like a Lewis A gene and a Lewis B gene. You just have a Lewis gene or not. So if you have a Lewis gene, you do step 1. You add a fucose and you make the Lewis A antigen. And then if you also have the secretor gene, then you do step 2. You add to the Lewis A antigen another fucose, and then you make the Lewis B antigen. So you make Lewis B antigen at the expense of Lewis A. You make Lewis B at the expense of Lewis A. And it's a stepwise process, so you could only make the Lewis B antigen if you have both the Lewis gene and the secretor gene, right? If you had just the Lewis gene, but not the secretor gene, then you would stop at Lewis A antigen, okay? And then if you ask the question, okay, what if you have no Lewis gene, but you just have the secretor gene? Then all that would do would be that you would add a fucose to your type 1 chain and you would have H antigen in your secretions. So you have H antigen in your secretions. If you happen to have an A or B gene, then you would further modify this to have either A antigen in your secretions or B antigen in your secretions. And then to summarize briefly, if you have no Lewis gene and no secretor gene, you make no Lewis antigens, mainly because you have no Lewis gene, right? You can't even start the process. If you have no Lewis gene, but you do have the secretor gene, you're still negative for Lewis antigens because, again, you have no Lewis gene. If you have the Lewis gene only but no secretor gene, you make Lewis A, just the first step. If you have both the Lewis gene and the secretor gene, you do both steps and then you make Lewis B. So you have positivity for Lewis B on your red cells. And that's all I have for red cell antibodies part 1. Remember to do the quiz questions to make sure you paid attention and learned something.